the call knows, this webinar will be recorded. So Janine with Alberta Environment and Parks, who's helping facilitate today, will help with the recording and the Q&A side of things. So thank you, Janine, for your help today. For those who don't know me, my name is Brad, and I'm the Executive Director here at the Alberta Lake Management Society. Obviously, we would prefer to be seeing everyone in person this year, but we're making the best of the bad situation that we have and inviting great speakers from across North America, folks who we otherwise wouldn't get a chance to hear from at our in-person conference. So I hope that everyone enjoys the webinar series. If you weren't with us last week, we had a presentation from Caleb Sin on the Alberta Lake Management Society programs this summer. And that was followed by a presentation from Nicole Kimmel with Alberta Environment and Parks. She's the aquatic invasive species specialist. And we did hear from Nicole that this year is looking to be a record year in terms of the number of mussel-fowl boats that have been stopped at our highway inspection stations. So if you're interested in those two presentations, they are now posted on the ALMS website if you want to check that out. That's just on the webinar page. Today we have two presentations this morning, one from Dr. Michael Rennie on microplastics in freshwater and one from Dr. Laura Johnson on the re-eutrophication of Lake Erie. So for the next couple hours, we'll be having presentations. Just logistically, the chat function is disabled right now. As well, viewers won't be able to turn on their microphones or videos, so there's no risk of accidentally interrupting the webinar. If you have questions throughout, please do just type them in the question and answer box, and Janine will assist with getting those questions answered, hopefully at the end of the presentation. So I think with that, we'll pass it over to Dr. Michael Rennie from Lakehead University, who will be able to now share your screen and start your presentation.
Okay, thanks a lot, Brad. I'll go ahead and do that now. Okay. So, is that sharing okay there, Brad?
Yeah, looks great.
Fantastic. Okay, well, thanks everyone for attending today. My name's Mike Rennie. I'm with Lakehead University and also a research fellow with the IISD Experimental Lakes area where I've been working since 2010. And I'm pleased to speak to you today a little bit about something that's small, but in size, but potentially very big in terms of the scope of the problem. And I think it's also something that I would say that we're still not fully able to grasp and that needs a fair bit of work to still figure out, and hopefully I'll be able to talk to you about some of the things that we have ongoing that will try and hopefully address some of those issues in the future. This could also alternatively be titled My Adventures in Microplastics. It's a bit of a story arc in terms of how I've been involved in microplastic research through the years and sort of where we're at now with some of the research that we have going on in my group. So to start off, I'm sure everyone's heard something about plastic pollution in the past several years. It's been in the news. We've got legislation that now exists in terms of limiting certain types of plastics from consumer products. For some old stats, just to give you some idea of what the extent of the problem is, back in 2014, they were estimating that there were something like 270,000 tons floating around in the oceans. That's probably quite a bit more now, with something like 5 to 13 million tons being added annually by coastal countries. So it's a lot of material. And part of the problem with that plastic pollution is that it breaks down, but it doesn't break down rapidly. So it tends to accumulate, and even the breakdown products seem to stick around for a while. So with all that plastic going into the oceans, where does it go? And a lot of what we know about is, it floats around and hangs out for a long time, but at some point it does seem to settle. It's most prevalent on seafloors. This is some pictures from, the bottom of the ocean several kilometers down, and we've got a package of, Uncle Ben's converted rice down there, as well as fishing gear. So, I mean, this stuff does sort of end up everywhere and in places that you might necessarily not think to find things like this. A large part of the concern over plastic pollution generally is also sort of what ends up happening with wildlife. So just to give you a sense of kind of what kind of impacts that can have, at least for the large plastic scale, I've got a few pictures here. This is an albatross chick from Midway Island, where you can see, the chick's dead, and you wonder why. Well, its stomach was basically jammed full of plastic. Seabirds in particular are pretty susceptible to this because they'll pick stuff up floating on the top of the water, bring it back to their chicks and feed it to their chicks, thinking it's food, but it's just, bits of plastic that's floating around in the water column. This is a shearwater from New Zealand. Again, the stomach contents of the bird is nearly jammed full of plastic. And when you see that large of a quantity of plastic in a bird, it becomes apparently obvious there's very little room for actual nutritive foods for room in the digestive tract. We also see it in fish too. This is a rainbow racer from the Pacific Ocean. A picture from the NGO organization Five Gyres. And, even we're seeing plastic in fishes as well, and not just in ocean environments, but as well as in freshwater. And that's what I'll probably talk a little bit about today. So I don't know if anyone's on Twitter, but this is a recent post that came out from the Beaverton, which is a satire news agency. But it doesn't take too long to think about how, with the onset of COVID since the beginning of the year, that we've got a large increase in the amount of plastic that's being consumed and put back out into the environment. So, face masks are sort of one piece of that, but you can think of, for a while there, you couldn't use reusable bags in grocery stores. Everything was coming in plastic from there. Fresh produce for a while, everything was being wrapped in plastic. Gloves, any kind of PP or personal protective equipment, unless disposed of properly, that can be a huge issue. And even when it is disposed of properly, we're putting it in bags that go to landfill. So the quantity and amount of plastic that we're generating is quite a lot greater. And it's not just speculation, so there's been some scientific publications to sort of demonstrate how things like face masks could actually be a much an increase or present an increase in terms of the amount of plastic that's being deposited into the environment, in this case in the form of fibers. So it's not just sort of like, yeah, anecdotally we're using a lot more plastic. There's actually some strong evidence now to suggest that this could ending up in the environment and potentially causing bigger issues. So what's part of the problem that we think about within the context of plastics? There is sort of this like macroplastic issue, which is the larger particles that end up everywhere, end up in the digestive tracts of birds. But then there's also microplastics, which, are far more ubiquitous. It's one thing to see, say, a plastic bag or a mask floating around in the parking lot of your favorite grocery store. But microplastics are something that every time we look for them, we find them in drinking water, in bottled water, in lakes and rivers, in air. One of the things that we have to do as researchers with microplastics is have controls set out in laboratory environments in the field, because this stuff is floating around all over the place. And we need to be able to know the extent to which we might have contamination from outside sources when examining our samples. So part of the concern around microplastics is simply because it really is everywhere. And we know it's not a naturally occurring substance, right? Plastics are a manufactured thing. Like we make it, and it's made in factories. And so if it weren't for humans, there wouldn't be plastics floating around in the environment. So we know it's an anthropogenically derived contaminant, let's call it. And so, and microplastics are sort of this broad category of plastics that are less than 5 millimeters. I'll just keep it at that for now. We can get into nanoplastics too, but let's just keep it simple. And you can break these types of plastics down into various forms. So we can think of fragments, which are just sort of these hard, jagged pieces, fibers, which seem to be pretty ubiquitous in the environment, and particularly in freshwater is what we see a lot of. We get films, which are sort of, you can imagine plastic bags, single-use plastics, like things that all your stuff comes wrapped in. Foams, so think polystyrene, you know, packaging for stereo and computer equipment and that sort of thing. And then pellets, which is a sort of a broader category, which could be anything from pre-production pellets to the kinds of abrasives we used to find in facial scrubs and that sort of thing before they were banned. So my microplastic story, I guess, starts with Lake Winnipeg. And when I was working with the International Institute for Sustainable Development, ELA, based in Winnipeg, where we had sort of started taking an interest in some research on Lake Winnipeg. So Lake Winnipeg is unique among sort of the large lakes in North America. It's the 10th largest lake in the world. What makes it really unique, though, is the fact that it's got such a huge watershed that feeds it. So it's about almost 1 million square kilometers that supplies the water to Lake Winnipeg, and that spans 4 different provinces, 3 different states, an international border. It's hard enough to regulate a water body when it's in one provincial jurisdiction. Imagine going across all these different borders, it becomes a challenge. Anyway, but within that very large watershed, you've got about 7 million people distributed throughout it, but that's fairly sparse. If you think about how that compares to something like Lake Erie, which is about a similar size, you've got something like 22 million people in a watershed that's, 4 orders of magnitude smaller. So it's not very dense, although it does contain several of the largest cities in Western Canada. And we got involved in this research largely just to try and get a baseline understanding of what's the extent of plastic pollution in Lake Winnipeg. If you think about what's around Lake Winnipeg, it's fairly, on the shores of Lake Winnipeg anyway, it's fairly sparsely populated. You've got some centers down in the south around Gimli. Along the whole eastern shore, it's fairly uninhabited. There's Grand Rapids up at the north where the North Saskatchewan River comes in, Dauphin, Manitoba. But there's not, and the Dauphin River, but there's not a whole lot of folks that are right on the shoreline. So in terms of plastic pollution in that lake, you might think it might be fairly low. Or if you do find it, then the idea is that it's got to be transported from somewhere else, right? So it's not like we've got a huge amount of industrial activity around the lake. So if we find it there, where is it coming from? So this research was done in collaboration with several people. So Phil Anderson, who was a summer student at ELA, was the lead author on the paper. He did a lot of the work to analyze the samples and do a lot of the counts initially. Sarah Warwick, who's currently a master's student at the University of Manitoba and now working with the IISD ELA, she did a number of the counts as well on this study. Victoria Langen, who was a past master's student in our lab, she did a lot of the microscopy work to try and confirm whether these particles were plastic or something else. Mark Hansen is a professor in the Department of Environment at the University of Manitoba. He was a supervisor of Sarah's and is a co-supervisor on her master's work. And John Chalice was at the time a PhD student at University of Manitoba. He's now a Banting postdoctoral fellow at the University of Saskatchewan. And he's been a big part of this research as well. And it ultimately resulted in this publication that came out in 2017 that describes the patterns of plastic pollution in Lake Winnipeg, and that's a little bit of what I'm going to summarize today. Though I should also mention, though, that none of this work would have gotten done without several other folks as well. That includes Karen Scott, who runs the research vessel Nemeo, which conducted all the sampling on Lake Winnipeg for 3 years. Karen is a huge advocate for the lake and has been instrumental in making sure that we understand that lake a lot better. Mike Staten and Hetty Kling sort of provided some of the anecdotal evidence for why we might want to look at microplastics in Lake Winnipeg, and I'll go through a little bit of that right now. So this sort of all started when I got some emails from Hetty and Mike saying, hey, you know, we're seeing plastics in Lake Winnipeg, we don't understand why. And so they sent some photos over, and this is what Hetty was seeing. So for context, Hetty and Mike, so Hetty is a phytoplankton ecologist. She does a lot of the counts for the lake, and she does a lot of contract work for other organizations as well. And so she kept seeing these little blue fibers popping up in her samples, which obviously aren't phytoplankton. And Mike sent this from some samples of whole water that he was looking at from around the Grand Rapids inflow from the North Saskatchewan, which kind of looked like maybe bits of monofilament netting, say from a gill net. Maybe it went through a turbine or something from a power generation plant. But we kept finding this stuff sort of floating around, but no way of kind of quantifying what it is that we have. And also no way of being able to compare, well, okay, if we're going to quantify it, let's make sure we quantify it in a way that we can make comparisons to other systems. So that led to sort of the initiation of this study, which was to pick several sites on the lake to try and establish what a baseline level is currently for plastic pollution in Lake Winnipeg, and employ methods that'll provide a means of comparison with other freshwater ecosystems. So at the time, most people were using these manta trawls to sample for microplastics. I'll go into some of the details there. And so the goal was to try and replicate the methods that were used on a study on the Great Lakes and apply it to Lake Winnipeg in order to make those numbers directly comparable. Okay, so that initiated the study in 2014, which ran for 3 years. We did fall sampling with a manta trawl. It's got a 300 micron mesh in order to make it comparable to the Great Lakes surveys. For anyone who's ever done any kind of like plankton or phytoplankton work, you're going to say like, wow, 300 microns is pretty big, Mike. You're missing a lot of stuff. I would agree. What I would say though is at the time, this was sort of what the typical sampling was, looking for surface plastics using this mesh size. So the main justification for using this method was to make the methods comparable and the numbers comparable that we generated to other systems. I would also say that there's been lots of work since that shows that this 300 micron number misses a lot of stuff. Oftentimes, if you look at sort of density peaks in terms of the microplastics that you collect from an environment, the peak densities usually seem to come out at around 100 microns. So we are missing a lot of stuff here, but again, it's just to make it comparable to other studies. We tried to mimic tow times and speeds to try and match other studies as well. And what ended up being modulated, you can see if you look in the sock at the back of the manta trawl there, it's pretty green. So if anyone knows anything about Lake Winnipeg, it's prone to algal blooms. And so what we had to do in order to make sure we didn't overfill the net because of collections of colonial algae and that sort of stuff was limit the amount of time that the trawl was in the net. Tried to keep the speed constant and everything else the same, but the time in the water is what ended up being variable to control that. Anyway, everything that was collected in the nets went into a glass jar and was preserved in 70% ethanol. So these are the sites that we sampled around the lakes. The numbers and letters are meaningless to you guys. What I would more stress here is point out some of the major inflows and outflows. The Red River down in the bottom part of the lake here is where most of the nutrients come into the lake. The Winnipeg River and the North Saskatchewan River, by comparison, supply most of the water to the lake. And the Nelson River up here, so let's see here. Lake Winnipeg flows into Playgreen Lake, which then goes up into the Nelson River. And then it ultimately empties into Hudson Bay. Okay, so what did we do? We used, again, to make methods comparable, we used wet peroxide oxidation as our treatment of the sample. So you can imagine drag a net through Lake Winnipeg at the surface for half an hour or an hour. These things were pretty green. We had all sorts of zooplankton in there. So the wet peroxide oxidation is to try and get rid of a lot of that biological material. So what you're left with are mostly plastics or other things that aren't plastics. From there, we use manual identification to pick things out from the samples. And then of the pieces that were picked out, we used a scanning electron microscopy with X-ray diffraction in order to try and tell us what the elemental composition of that stuff was. So we could easily say, oh, this is a paint chip versus, or a rust chip off the boat versus this is an actual piece of plastic. Okay, so what did we find? I'm just showing you results for 1 year in terms of the spatial distribution of the density of plastics around the lake from those sampling sites I showed you earlier. This is all in Phil's paper from 2017 if you want to go and look up the full details. But basically we found plastics sort of throughout the lake. We didn't see any real patterns in terms of them being more or less concentrated in the north or south basin. And the counts were fairly consistent between years. There were no significant differences anyway, if we looked at the counts sort of across time. So, on average, we found that over all 3 years, the densities of microplastics that we were finding were about 200,000 per kilometer squared. So you're like, well, 200,000, that sounds like a lot, right? But if you put that down into meters squared, that's 0.2 particles per meter squared, right? So, scale that up to 1 particle for every, whatever, like 10 meters squared. So that it's a number, but what we really need to know is how does that compare to other systems? So we were able to take a look at that 2014 study from the Great Lakes and put our Lake Winnipeg data in as comparison to see how they compared. And surprisingly, we found concentrations of microplastics in Lake Winnipeg using the same sampling methods and the same processing methods that were comparable to Lake Erie, which has 4 times the number of people or 3 times the number of people living in the watershed, much more densely, a huge amount of industry and industrial activity that we don't have in the Lake Winnipeg watershed. Most of Lake Winnipeg is in northern Manitoba, where there's really not a lot going on. So and much higher concentrations than we saw in either Lake Huron or Lake Superior from that same study. So this was surprising to us. Why would we see in a lake like Lake Winnipeg, which really isn't very close to anything at all, such high densities of microplastics comparable to the Great Lakes, and in some cases even higher compared to either Lake Huron and Lake Superior? So if you took that density of 200,000 per kilometer squared and scaled it up just to say, okay, at any given time, how many microplastic particles that are greater than 300 micrometers are we likely to find in Lake Winnipeg? That number works out to about 4.8 billion. What kinds of plastics were we finding? So the vast majority of the plastics that we found in Lake Winnipeg were fibers. So this was sort of the main thing we found there. And And it's interesting when you look into the literature and see, well, what's the kind of plastic that you see that's often deposited atmospherically? So like if you set out a bucket and put it up on your roof for a few days and see what shows up there, it's mostly fibers. So when we think about the Lake Winnipeg situation, either we've got a case where things are being transported for very long distances down riverine systems. So I don't know, is it coming all the way from Calgary or Edmonton, Saskatoon, and then, all the way into Lake Winnipeg, or is it atmospheric deposition? Those are still questions that I think are unanswered within this context, but we're doing work at ELA that I think is going to help inform some of that. So the next step that we did, so Sarah had sort of done all this just for fun through her undergrad, and then decided to do an undergraduate thesis in the Department of Biology, and decided, okay, well, we know what's in the lake now, so let's take a look at a couple of the river systems around Lake Winnipeg and try and get a handle on the density of microplastics there. Because if we know what's in the lake, it would be nice to know what's coming into the lake and what's leaving the lake, so that we have some idea of, how does that loading compare? Does stuff come in and what comes in comes out? Or is there evidence of some kind of settling or accumulation in Lake Winnipeg? So this was the sampling design that Sarah came up with. There was, they did sampling way down at Emerson at the US-Canada border here. They sampled sort of before and after the South End Pollution Control Center, which is a water treatment plant in Winnipeg. They sampled at the Forks, which is after the West End Pollution Control Center, and then sampled up here at the Royal Manitoba Yacht Club, which is after the North End Pollution Control Center. And then I think Selkirk Park, but I'm not sure if that shows up in these data. And then later in the summer, big trip up to the Nelson River to do some sampling there to give us some sense of what's coming out of Lake Winnipeg. And this is all data that was published in a paper that came out with Sarah as the lead author in 2017. So here are the densities of microplastics that were found in the river. Again, this is sampling with a manta trawl at different sites and different time periods. Overall, what we would expect, if we think about that map that we just looked at, we'd expect to see an increase in microplastics as we go from Emerson to the Royal Manitoba Yacht Club, which we kind of see. There's a really low number at the Royal Manitoba Yacht Club back in, what is that? That's going to be like July 2016. But on average, if you looked sort of downstream of treatment plants versus upstream of treatment plants, we did see a higher number sort of downstream from those treatment plants. What's confusing though is if you sort of work the Forks into the middle of this, this is where the Assiniboine River meets the Red River. That's actually in between the Redboyne and the Yacht Club sites. So we had this one really high density count there in June of 2016, but upstream of that, we didn't see a much larger value. Anyway, so generally, there's maybe a little bit of evidence here that we're seeing some contributions from wastewater treatment plants, but not convincingly. And I've seen other papers from other groups that have started looking at this in other parts of Western Canada, and we're not seeing huge spikes upstream or downstream of treatment plants. So that but we're still seeing them. So again, where are they coming from? It sounds like this is starting to becoming more of a picture of these are non-point source contaminants. And then if we look at the Nelson River, which is the outflow of Lake Winnipeg, where we know there's 4.5 billion particles floating around in the lake at any given time, we see a very low count. So we tried to estimate flux of particles. So we took the number of particles in the water column, adjusted it to a volume, used a cross-sectional flow to try and understand how much of that plastic is moving by at any given time. And then we Z-scored those values so that we could compare across sites to make things statistically comparable. what this showed was that we did seem to see a very seasonal pattern in flux, which is maybe not surprising, right? Especially anyone who's ever been to Winnipeg knows that in the spring, we have very high flows and very high lots of water moving through the city. So to show that plastic is sort of scaling with the amount of water that we've got moving through the system is maybe not surprising. What was interesting is that we did see an interesting correlation between the flux that we see in the river along with the flux that we see at the discharge from wastewater treatment plants. So when we have large amounts of discharge from the treatment plants, we saw greater flux of microplastics in the system as well. So, and these are all data points from within the city of Winnipeg. But again, the seasonal pattern comes out where these high points are all in the spring, but all these low points down here in the fall. So, and if you're in a system where you've got blended water treatment, so not all of your stormwater and your municipal wastewater are not always separated, which is the case in Winnipeg, then maybe this correlation makes sense. Okay, so how much is being contributed by the Red River? So that we can say that 4.8 billion was sort of is a constant number that we might see in Lake Winnipeg. It's, you know, comes from an average over 3 years. The estimated annual input that we got from Sarah's work is about, 400 million or 0.4 billion per year coming in from the Red River. But if you look at the losses to the Nelson River, we're only losing about 4 million or 0.004 billion per year. So if the amount of plastic in the lake is constant, it's not going down, then what that means, and by the way, this doesn't even think about anything coming in from either the Winnipeg River or the North Saskatchewan, which is where we know most of the water comes from in the lake anyway. So if that value doesn't change much from year to year in the lake, but we're seeing far fewer particles come out of the outflow than from what's going into the lake, then it suggests that, well, initially, that 99% of the stuff that's coming into the lake is unaccounted for. So where is it going? So our working hypothesis is that it's sedimentation, but we really need to go out and do some sediment grabs and try and understand sort of what are we seeing in the sediments of Lake Winnipeg in order to understand if that's actually what's going on or not. Sarah also did some work to show that fish were accumulating particles as well. So this was sampling that was done in the Red River around the city of Winnipeg and found counts on average of about 7 microplastics per fish in something like 30 carp that she sampled. So, so they're accumulating it, but it still sort of begs the question, like, how is that going to have an impact on what fish grows and reproduces? And that's one of the things that we're not entirely certain of still. So to summarize our Lake Winnipeg work, we know it's comparable to other Great Lakes, that most of that pollution is coming from fibers. We did see a seasonal pattern in terms of the loading that we saw there, and some evidence, we think anyway, of retention in the lake. So I want to switch gears in the small amount of time I have left to try and just highlight some of the ongoing work briefly that we're doing here in my research group. So that involves so now that we've seen some evidence of settling in Lake Winnipeg, trying to do some experimental work to try and understand what actually drives settling rates of microplastics and trying to understand the impacts of nutrients. Some background microplastic sampling that we've done around ELA as part of a much broader project, which I'll talk about in a second, which also is sort of geared at trying to understand the behavior of microplastics in natural environments, as well as understanding some of the potentially sublethal responses of microplastics when exposed to fish or other organisms. So the settling work that we're evaluating is part of Sarah's master's research. So Mark Hansen has all these huge cattle tanks that are set up out on a field outside the University of Manitoba. And he's done a lot of contaminant work looking at wetlands in these things. So we were able to take advantage of those to try and look at some both short-term and long-term patterns of settling of microplastics to try and understand sort of, you know, do we get resuspension? What are some of the overall dynamics that we see? So over almost a 2-year period, we were able to run our first set of experiments, trying to see if we got any evidence of resuspension sort of with freezing and then thawing in those systems. And we don't seem to see a whole lot of evidence for that. What we do see is a fairly sort of constant decay rate for certain particles. Fibers and films seem to be the things that took the longest amount of time to settle out or actually did something over the time period of the experiment. So fibers seem to settle out fairly quickly over about a 28-day period and then just sort of never really came back into the water column, as opposed to films which stayed in the water column for a very much longer time and then settled out sort of, it took looking at this sort of a year or more to settle out of the water column. And then Sarah also did some interesting work in the second year to try and understand, are there certain factors that are going to encourage the deposition of microplastics? And while she's still working this up as part of her thesis, her initial findings seem to suggest that at least for particular substrates, so this is again sort of looking at both fibers and films, that at least for fibers anyway, it seems like the addition of nutrients seem to encourage the deposition of those fibers out of the water column at a faster rate than we saw in the control tanks. And then also because these are sort of like wetland simulated mesocosms, ones that had emergent vegetation also promoted the settling of microplastics out of the water column. So it's starting to point to some of the things that might actually encourage sedimentation of plastics in natural systems. And if you think back to the Lake Winnipeg study, where we know we've got a lot of nutrients coming in from watersheds, agriculture in the south, that may speak to why we see or have evidence of potentially high amounts of settling in that system. So the last piece I want to talk to you about briefly is the plastic project, P-elastic project. So this is a project that I'm involved in that we've now been sort of developing for a few years to try and understand the fate and effects of microplastics in aquatic ecosystems at the experimental lakes area. And the two real sort of main things we want to try and do here is to understand the impacts at environmentally relevant concentrations of microplastics in natural systems. So we've seen lots of lab studies in the literature where if you take some Daphnia and put them in a vial that's half plastic, that they don't do so well. And that's not surprising, but that's not what we're seeing in the environment currently. So in our experiments here, we're trying to understand what are the impacts of plastics at the whole ecosystem level within the realm of something that we might expect to see within the next 50 years, right? We're also trying to evaluate both, because we're doing these at sort of the whole ecosystem scale, it's allowing us to evaluate both direct and indirect effects on ecosystems. So indirect effects are sort of one of these things where if one organism responds, but not the rest of the experiment to being exposed to, say, microplastics, well, the response of that one organism can actually cause cascading effects throughout the ecosystem. And we've seen that in several experiments at ELA in the past. So this is sort of the cast, the plastic cast. All of the principal investigators that are involved here. I'm just going to highlight Chelsea Rockman, who's sort of the lead. She's at the University of Toronto, probably one of the leading researchers in terms of microplastics and plastic pollution in North America, if not the world. And Diana Rehill, who's at Queen's University, who's done a ton of work out at ELA in the past. And then that's supported by folks both in government, so at the Ontario Ministry, or sorry, now it's the Ontario Ministry of Environment and Parks and Environment and Climate Change Canada, and folks from across the US and Canada involved here. We've also got about a dozen graduate students that are involved in the work here that aren't listed on this slide. So we really have, I feel like, the crew of people that are going to be important in sort of driving this work forward. Here's the timeline for our experiments. So we've done some reference sampling, background sampling on ELA lakes to try and understand what sort of is out there with us doing nothing. We're doing some background sampling on an experimental lake where we're going to set up some limnocoral experiments, mesocosms, to try and sort of do a range of concentrations to understand what that looks like. And then ultimately that's all going to inform a whole ecosystem experiment, which we hope to get going in 2022. I'll highlight some of the background microplastic work here that's been done primarily by 2 undergraduate students. One is Haley McIlwraith at University of Toronto, and another is Nadine Elmaricki at Lakehead University. So this is just a map of ELA and all our lakes. You'll notice that we use numbers, we don't use names because we have a lot of them. Anyway, so Haley used a peristaltic pump to sample plastics from the water column to get counts. And I'll just show you quickly some data here. Originally, this design was set up to try and understand, okay, in lakes that get a lot of canoe traffic and people going through them, do we see higher plastic concentrations in lakes that have more sort of human activity on them? We also looked at lakes that we do a lot of work on versus ones that we don't go to all that much, right? Because any lake that we work on, it's got a dock, it's got center buoy set up, that's got a rope in there. So there's obviously sources for plastics. And what was interesting is that, you know, across this gradient of activity, human activity, we really didn't see any sort of difference in microplastic concentrations that correlated with that activity, which again sort of points to this. Well, if it's not us, what's contributing to plastics on the landscape out here at ELA? And we did see, I mean, we do have atmospheric deposition out there. So this is from some atmospheric collectors that we had set out, and we're seeing plastic deposited from the atmosphere. We need to take this to the next level and try and do some mass balances to see how much atmospheric deposition is contributing to the concentrations that we're seeing in these lakes. But, you know, it's pointing to some potential sources here that aren't necessarily just anthropogenic in terms of like us being physically on the lake. Again, we did some sampling with a manta trawl. So this was Nadine's work. Just did 3 transects across the lake as part of a field course that she was participating in. And this is a comparison, again, using the same methods, comparisons to other lakes. Again, Lake Winnipeg and Lake Erie sort of take the cake in terms of the mean concentrations. I'm going to zoom in here a little bit and just ignore those really high concentrations. But if you look at Lake 224 and Lake 378, these tiny lakes in the middle of nowhere still have higher concentrations of microplastics on a per meter squared basis than we're seeing in either Lake Superior or Lake Huron, which, yeah, that's surprising. So stuff that we want to think about maybe a little bit more. This is a picture of what we think the mesocosm study is going to look like, right? So this is actually from a work that's being done on Lake 260 at ELA, where they're evaluating the impacts of diluted bitumen on aquatic ecosystems. It's all mesocosm based, which is why they have all these berms set up in the lake in case there's a spill or something like that. These pictures were all taken before any oil was added, but you get a sense for sort of the scale here. So we're setting up mini ecosystems within a lake to try and understand how a range of or a gradient of plastic concentrations are affecting sort of these mini ecosystems within the lake. And that's going to help us inform what we're going to do at the whole lake scale the following year. As part of that mesocosm experiment, Cody Veneruzzo, who's a master's student who's just started at Lakehead, is going to be looking at the respirometry of fish. As we take the tanks down, all of those 10-meter mesocosms are going to have yellow perch in them. We're going to be looking at the potential for respirometric impairment in those fish and trying to relate that to the growth rates of fish in those mesocosms through the time period of exposure. We're also trying to look at any potential, because the walls of the mesocosms are plastic too, in a plastic experiment. We're doing some work right now at the Aquatic Toxicology Research Center at Lakehead University, just to try and evaluate whether there's any toxicity potentially associated with the mesocosm walls that we plan to use. And that's being undertaken right now also by Cody. The last thing I'll highlight here is some work that's being done by a PhD student in my lab, Haley McLeod. She's actually going to be using the range of concentrations that we're applying in the mesocosm experiment to try and see if that has sublethal effects on the, and lethal effects on the development of lake trout eggs. So we're getting some eggs from the Dorian Fish Hatchery just down the road. And this is a preliminary setup that she had going last year. We'll have about 10 times the number of tanks set up in this cooler room here to try and look at how the development of both the eggs and the larvae for several months might be impacted by the exposure to microplastics. Ultimately, this is all hopefully going to inform a whole lake experiment, and the part that I'm going to be involved in there is trying to look at sort of fish responses at the population level, individual and population level. So looking at growth rates, looking at body condition, looking at population densities, which is some of the great work that we can do at ELA with the mark recapture programs that we have in place that you can't do a lot of other places. So I will, in the small amount of time I've got left, so hopefully we've got time for questions. Just want to say, you know, if there's anything that I've hopefully been able to show you today, it's that microplastics are everywhere. I mean, they are where we look for them, and that seems to be the case with every other paper that comes out every week. Where they're in the Arctic, they're in the most, you know, at the tops of mountains in Alberta. They're in the most obscure places you would imagine to find them. Bottled water, tap water, they're all over the place. But just because they're everywhere doesn't necessarily inform how much we should be worried about it, I guess, right? I mean, it's not, we could say it's not natural, and I would prefer to drink water with plastic in it. But is it having harmful effects? And I don't know if we have enough information to make that assessment yet. And so what we're really hoping is that the work that we've got planned out at the experimental lakes area is going to help us address how much we need to worry about this, particularly at the whole ecosystem scale, which can ultimately guide some environmental policy on how we deal with microplastics moving forward. With that, I'd just like to acknowledge the following funding sources for all their assistance and support through the years for all the projects I talked about today, and hopefully we've got some time for questions. Thanks.
Okay, thanks so much for the presentation. That was great. It's always terrifying doing it over Zoom because we're worried it's going to like crash or something, but it was perfectly smooth on my end. So thank you so much for your. Yeah. Janine, I think we have a couple of questions in the chat.
We sure do. Thank you, Michael, for that great presentation. That was super interesting. So our first question is, do you have any plans, or is there the possibility for paleo work for microplastics in Lake Winnipeg?
We don't have any planned. What's interesting though, there are several cores that exist from Lake Winnipeg. I believe they're all with Peter Leavitt at the University of Regina. And there's been some papers that have come out from those cores, but it certainly raises an interesting opportunity as to whether, if there's sample left, one could sort of dig through there and see if there's any evidence of deposition through time. I guess the challenge would be the scale, right? So if you think about, you know, 1 particle per 10 meters squared, is a core that's only got a 3-inch diameter going to be enough to be able to give you any kind of actual, you know, are we going to see enough? I'm not sure. So, so I think there's some good opportunities there to take a look, whether sort of a standard coring methodology is going to do it, or if we need something, you know, bigger. Maybe we should use like Ekman dredges or Ponars as a start to see what the surface concentrations are like, and then take it from there. I'm not sure, but yeah, we had talked about doing some sediment work back when this was sort of more active. The tough part is trying to convince someone to pick through sediments to look for plastic particles. It's not an easy job, so.
Okay, great, thank you. And our next question, part of which you already answered through your last slide, but is there any involvement from ECCC or DFO on your project?
Yeah, so the National Contaminants Advisory Group is a big financial supporter for the research that we've got moving forward for the mesocosm work, which is great. So what I would say is, if you recall, back in 2012, which is also the reason that I no longer work at ELA as a federal government scientist, is that DFO is no longer in the business of doing active research on contaminants. They fund it through the Contaminants Advisory Group, but they're not actually involved in that work. So we're lucky to be involved with DFO at the level that they are now in this kind of research. On the Environment Canada and climate change side, yeah, Jen Provenche is one of the members of the research group that we've got going for the plastics experiment out at ELA, and she's got a massive background in this and is sort of like tied in pretty heavily to all that. We've also got Paul Helm working with us from the Ministry of Environment Conservation and Parks in Ontario. So we've got multiple levels of government involved, and so and with our work with IISD, I feel like we've got all the pieces in place to be able to quickly translate what we find into policy that government can use.
Great, thank you. And we've got another question here. Is there a community that is active to help with the next project?
Well, yes. I mean, so I think we've got what, like 15 PIs on the current work that we've got planned out at ELA. Several years back, we did put in an application. So as a follow-up to the Lake Winnipeg work, we had an application in with Environment Canada to try and look across the watershed of Lake Winnipeg and see if we could try and understand what kind of long-range transportation we were getting through some of the major tributaries versus atmospheric or something like that. That work wasn't funded, but I do know that several of the groups at different institutions are now starting to publish work. They were able to do that work independently and are now starting to publish some of that stuff. So that's starting to come out in the literature now, which is kind of nice to see. And some of that, one of the things that we had tried to build into that was some community-based sampling, but I'm not involved with any of that currently. Chelsea Rockman in Toronto has done some really good sort of community-based stuff that's actually resulted in data collection and papers too. So that's encouraging to see.
Okay, great, thanks. And then I actually have a question for you as well. Sure. Have you guys done any consideration in terms of the invasive mussel population and accumulation of microplastics in those in Lake Winnipeg?
Not really. It would be interesting to see. So yeah, so as someone who's done work on the Great Lakes, my PhD was I did a lot of work looking at sort of impacts of dracenids on corygonids and whitefish particular. One of the things that we often struggle with is having data before the mussels show up. And even in Lake Winnipeg, it was one of those things where, we've got a fairly good idea that they're coming, right? We should really get some background data. And so trying to generate funds to get that data before you know there's a problem is sometimes a challenge, right? There's certainly evidence that dracenids, through their filtering capacity, have pretty dramatic effects on algal community composition, on pathways of nutrients in terms of how the energy shifts from sort of offshore to nearshore processes, particularly in the Great Lakes. There is certainly a possibility that they would contribute to sort of the removal of these things, but it's hard to say. So yeah, it would be interesting to know. Some of the background data that we have on Lake Winnipeg would potentially be useful for this, because all that data was collected sort of right as they were starting to establish, so in very low numbers, and I would say unlikely to have had any ecological impact on the system. So maybe if we repeat that study in another 5 to 10 years, we might actually be able to answer that question.
Great, thank you. So we just have one question left here. So are you considering the potential for microplastics to act as a vector for pathogens in your whole ecosystem experiment?
Potential for that's a good question. I don't know that we've got anyone explicitly looking at pathogens. We do have, like, we've got people that are looking at sort of microbial communities generally, I think. But I don't know that we've got anything specifically geared towards pathogens. That's a good question.
Okay, great, thank you. That's all the questions that we have that have come in so far. Again, thank you very much for your presentation. That was really great.
Happy to provide it, and thanks everyone for showing up today.
Brad, you're muted.
Thank you.
There we
Okay. Thanks, Dr. Rennie, for your presentation. And we have our next presenter here. Perfect timing. Thank you for, it's just so seamless. I don't know how these things work out. So we have Dr. Laura Johnson, who's joining us now for our next session. You should see at the bottom of your screen that I think you have the share screen ability. I'll just run over some logistics as we get that set up. So for those who are kind of just joining us, the chat function is disabled, but you can submit questions through the Q&A feature in Zoom. At the end of the presentation, we can facilitate answering those questions. We do expect to have our next speaker for about the next hour or so. So feel free to send the questions in as you think of them, and toward the end, we can help work through those. Dr. Johnson, if you run into any troubles or technological issues, of course, just give me a shout and we'll work it out. Seems to be working okay so far.
Great, so you see my screen and you can hear me.
We can see your screen, you sound loud and clear, so that's great. So I think what we'll do, we'll just jump right into your presentation if that works for you. And I'll let you kind of introduce yourself. I'll just say I was looking for a presentation something to do with the Great Lakes, because we have this ability through the online format this year to bring in new speakers. So I'm so grateful that you were eager and interested in giving a talk to us here in Alberta today. So take it away.
Awesome, thank you. All right, I'm going to move this. All right, so hi everyone, I'm Laura Johnson. I'm director of the National Center for Water Quality Research, and that's located at Heidelberg University. And the Heidelberg University I'm talking about is in the middle of nowhere, Ohio in the United States, not over in Germany. So I do get that every once in a while that people think that we're actually located somewhere else. And I should have had the slide that had the map on there so you could see exactly where we're located, but I'll point it out in a couple of slides here. And at the NCWQR, we've done a lot of water monitoring research, and so I wanted to share some of that with you today and how that links to issues that have sprung up in Lake Erie. I should have a disclaimer, however, that this is very regionally focused. I think that I have some good takeaways for you guys to be considering and some of the issues that you might have, but when it comes to very specific agricultural practices or types of runoff that you might see that might be slightly different, and I probably know very much less about some of those specific issues you might be dealing with. So to start, I just wanted to show a picture. This is an aerial photograph of the western end of Lake Erie in 2019. And you can see this is showing a state park. This is Maumee Bay State Park, and that has a beach. There, I have a little zoom in of what that water looked like. This is in August of 2019. And you can see these blooms get really big and really scummy. And, in some ways, kind of like on such a large scale, they're rather impressive. I do have an inset showing that although we always talk about algal blooms in Lake Erie, we're really talking about blooms that are occurring on the most western end, which is not even a whole full third of the lake. So it's a shallow, warmest end of the lake. Now, we know that blooms need a number of things in order to occur. Light's the first obvious thing because we're talking about things that are photosynthesizing. What blooms really need are warm temperatures. And so the time of year in which we see blooms occur on Lake Erie is fairly limited because it has to be when water temperatures warm up in the summer and are warm enough to support a bloom. And then the third thing is nutrients. Being that we're talking about the shallow end of Lake Erie that does warm up rather quickly, the first two things are things that are usually not too limiting once we get into summer. And so what I wanted to talk about in my presentation today is more of the nutrient side of things. What I didn't mention, and I probably should have mentioned here, is that a quick reminder that these are cyanobacterial blooms. I'll call them, harmful algal blooms. It's microcystis. It creates a toxin called microcystin, which is very harmful to human health and leads to some big challenges. But algal blooms in Lake Erie are not a new thing. They were quite prevalent in the 60s and 70s. And with the limited data we have, and then lots of observation, they seem to have improved as we moved into the mid to early 90s. You can see there's some spotty data here, but then they've come back with, a plum since the 2000s. And that's when we started to see these really big microcystis blooms that are forming. And not every year is big, but we do have some very intense years. If we focus in just on the 2000s, NOAA was able to develop, using satellite imagery, to assess bloom size and put a bloom size index on here. And you can see the variation in bloom sizes. But the one thing is that we've had some really big blooms, especially since 2008. I've pointed out here in 2014, when there's the Toledo drinking water crisis, where the microcystin toxin was found in the finished drinking water in Toledo. And so drinking water was, essentially banned for a weekend until they got those concentrations back down. Luckily, over this whole period of time, much of this period of time, we've been monitoring a lot of these tributaries going into Lake Erie as a part of our tributary loading program. Currently, we're monitoring 23 different locations, and this is the state of Ohio. So you can see we're focused primarily in Ohio. We do have one station up in Michigan because it goes into Lake Erie. Our very longest term stations are the Maumee River and the Sandusky River, where we've been monitoring there since 1974, which means we are in the middle of our 45th year of sampling on those two rivers, at least in terms of water year. We added, shortly after the Maumee and the Sandusky, we added like the River Raisin and the Cuyahoga, because those are also two big inputs going in. A couple of small watersheds around the Sandusky. These three rivers that are in the south part of the state go down towards the Ohio River, so they don't actually go to the Great Lakes. And then we have three stations around an inland reservoir in Ohio called Grand Lake St. Mary's, which I'm going to touch on a little bit, even though I promised to only talk about Lake Erie. I can't help it. So you'll hear a little bit of that too. At each one of these sites where we're collecting water quality data, the USGS is basically our Environment and Climate Change Canada when it comes to the water monitoring side of things. And so they're taking care of all of our water quantity measurements, like how fast is the water moving, how much water is there. Our newest stations that we've added are like Shallow Run and Potato Run, and they're there to help us be able to test if we change practices on the ground, how much of an effect will it have. So if you went to any one of those red dots on the river, oh, I should have pointed out, sorry you all, the Rock Creek here in Tiffin, this little dot, that's on campus. So that's exactly where I'm located right now. I can see Rock Creek from my window. Okay, so if you went to any one of these red dots, essentially what you would see is a station beside a creek, usually up on stilts, that has there's a pump in the river that's essentially continually pumping water through up into that building, and it's draining through. We then sample that water with a refrigerated automatic sampler, and that pulls a sample 3 times a day all year round. So when I sat on the Maumee and the Sandusky, we started sampling in 1974. We never stopped sampling either. We basically started, there was like bridge construction, so we missed a little bit here or there, or if like the pump breaks, we missed a little bit, but we have very consistent data collection. Every Monday, our technicians drive around, collect the samples, bring them back to the lab, and we analyze them for all sorts of things. Various forms of phosphorus, various forms of nitrogen, the amount of suspended sediments in the water, and then some other interesting things like chloride and sulfate, and those things can tell us about weathering and like road salt runoff. We also do some pesticide monitoring at some of our stations. And so what this has done is it creates these huge continuous nutrient concentration databases that are sometimes a bit overwhelming to comprehend. You can see this as an example of one of the analytes from one of our rivers. Of course, I chose the Maumee because that's what I'll be talking about today. But what we analyzed for is actually 11 different things from 23 stations right now. So you can imagine we have a lot of data to work with. So let's answer the first question, and that is that why did blooms return to Lake Erie in the first place? And to answer this question, I want to zoom in on the Maumee River. And this is basically a map that's showing loads. This is for total phosphorus coming from all of the major tributaries going into Lake Erie. The size of the arrow is related to the size of the load. And the very largest arrow you see is the Maumee River right here. The next largest arrow is the Detroit River, but that's actually bringing a lot of the flow from the upper Great Lakes. So this is really just an interconnecting channel. And the loads are about the same as the Maumee, even though the Detroit River is bringing 95% of the water into the Western Basin, and the Maumee River is bringing a little bit less, only 5%. The Maumee River then is highly concentrated in the nutrients that it's bringing in. So we're going to focus in on the Maumee River, and I want to show you some of the long-term trends that we've found in nutrient concentrations. So oftentimes we think of nitrate as one of the things that can control blooms. And this is showing annual concentrations of nitrate, nitrogen. Each dot is a different year of measurement. And then I have just the 5-year running average going through this so you can sort of see what the trends are. I also marked on here at the bottom the general time periods in which we had blooms that kind of went into the mid-80s, when blooms basically went away unless there was really, really high flow year. I think there were some in the mid-90s. And then in the 2000s when blooms returned. And what you notice, I think, hopefully, with the nitrate nitrogen here is that the patterns, the trends in nitrogen concentrations are actually, they're kind of exactly opposite of what we would expect if it was nitrogen that was controlling the blooms. It's lower when the blooms are here, it increased through the 90s, and we saw a pretty precipitous decrease in the early 2000s that we sort of leveled off from. We also look at particulate phosphorus. So this would be if we took our water sample, you can see a sample here, and we filtered it, the particulate phosphorus would be what was left over on the filter. Phosphorus is really sticky, so it likes to hold on to sediment like that. And what you see for particulate phosphorus is it has this basically steady decline from the 70s through to the 2000s, a little bit of a change here in the middle, but mostly no real change over the past 10 or 15 years. This makes sense because a lot of the early phosphorus control was trying to reduce phosphorus loss from fields, basically from particulates by controlling soil erosion. And you can see it was pretty effective actually, but doesn't correspond to our changes in blooms. The thing that does correspond with the observation in blooms is dissolved phosphorus. So this would be if we took our water sample, filtered it through the filter paper, and whatever was left over, whatever you can't see that made it into that water sample, that's dissolved phosphorus. We care about this because it's really bioavailable for algae to use. It's like the sugar of the phosphorus world rather than like the grape nut cereal, right? So dissolved phosphorus was high in the 70s, decreased into the 80s, increased through the late 90s, and has since remained high. And this corresponds really, really well with our observations with changes in blooms. So a lot of our efforts and thoughts go into, well, we have this dissolved reactive phosphorus issue. How do we control that? But one of the big questions is, well, where is this dissolved phosphorus coming from? And one of the best ways to do this is to take a watershed approach and then try to do a mass balance of the nutrients to say where they're coming from. So I like this particular watershed diagram because it covers most of the things that I care about oftentimes. So let's say we're monitoring down here at the end of this watershed, and you can see we obviously have some farmland, we have some residential area, we have a city and some forest, and then we have two other things. We have septic tanks that are down in the one corner, and then we have a wastewater treatment plant in the other, right? And so those are all of these main sources of phosphorus when we think of a watershed. We can have runoff coming from the land, non-point sources, or we can have these point sources like wastewater treatment plants. And then usually what gets forgot about are the septic tanks. And so we can attribute phosphorus to each one of these. And this is an effort that was done by the Ohio EPA. So they're the ones that do all the permitting for discharges in our area. And you can see that they're able to, because they get the permitted data, they're able to assign a load to things like wastewater treatment plants or industrial inputs. You can see they've done estimates here as well for septic tanks. And we have it for both phosphorus and nitrogen. This is specific to the Maumee River. What should jump out here is that really all of our phosphorus is coming from non-point sources. And essentially that means it's stuff that we can't attribute to. It's all land runoff. I should have mentioned that these permitted discharges also include estimates for combined sewer overflows. So I don't know if you guys have any issues with that in your region, but that's incorporated into this estimate. So with the land runoff, you know, when we go to then say, well, where could this be coming from? If you drove around our landscape, it looks kind of like this. We have an awful lot of very intensive row crop agriculture, primarily corn and soybeans, sometimes a little bit of wheat. So we attribute this to agriculture because that's what our landscape is. But what we finally learned over time as well is that it's not just these long-term trends, it's not what's happening over an entire year, but the actual very specific timing of dissolved phosphorus loading is really important for dictating bloom size. And part of this came from some observations that went back to around 2011, 2012. So I'm showing you here the bloom severity index. You can see the satellite imagery of these 2 years. And 1 year we had these huge spring rains in May. Then that was in 2011. And then in 2012, it was essentially a drought, like coming right out of winter. It was really warm and super dry. And you can see on the bloom severity index, we went from a really high bloom year to one of the lowest we had seen in the past 4 or 5 years. So really high to really low, right back to back. When we looked at our annual dissolved phosphorus load, or even just stream flow discharge coming out of the Maumee River, we didn't really see that big of a difference between 2011 and 2012. And it turned out it was because in 2011, all the rain came in the spring, and in 2012, it was in the fall. And so if we narrowed our window to the springtime, and we extend our spring all the way to July, because it's important for this bloom season, you can see that's where we start to see that really big difference between 2011 and 2012. So if we take all these data and relate them to each other, we end up with a relationship that looks something like this. So this is total bioavailable phosphorus. So it's basically all the dissolved phosphorus, and a very small portion of the particulate phosphorus that's actually available ends up being about 8% from March through July, versus the size of the bloom ranging from around 2012, which you can see is a fairly minor bloom. This is the actual intensities, those CI intensities that are produced from the satellites, up to 2015, which was our largest bloom to date. And in 2015, the bloom extended out of the Western Basin, was eking over towards Cleveland. So this is the relationship that builds the basis of us being able to forecast bloom size each year. So I want to talk a little bit now about 2019, because 2019 was a really weird year for us. So you can see this is that same figure, but now I've added in 2019 here on the end. You can see that stream flow discharge, so basically the amount of water coming out of the Maumee River, was the third highest that we had in this figure. Ended up being the third highest that we've had for the whole time we've been monitoring the Maumee, which goes back to the 1930s. USGS has been monitoring, I should say. But our dissolved phosphorus load, although it was high, it wasn't extraordinarily high. It wasn't record-breaking high. In fact, if we had looked at flow, we would have predicted that dissolved phosphorus loads would have been 30% higher, so somewhere up here, rather than where it ended up being. But because we use total bioavailable phosphorus instead of either just particulate phosphorus or total phosphorus or flow to do the forecast, what we ended up forecasting was not the highest bloom we'd ever had, but instead a 7.5. And what we ended up observing, you can see there's a big range here because we use, there's multiple models that are used to develop these forecasts. We ended up observing was a 7.3. If we had used flow or total particulate phosphorus, we would have expected something like 2011, because the flow was almost exactly the same as that year. Okay, so the big question here is, well, why was 2019 all that different than 2018? Well, in our area, we got rain, a lot of rain, not like, you know, gully washing rain, flash flood rain. It was just like very consistent, and essentially made it that's from the fall before all the way through the winter and into the summer, farmers just could not get out on their fields. They couldn't drive their equipment in their fields because it was too muddy, they would get stuck. And so this is an image that's showing vegetation index between 2018 and 2019. And you can see the delay in planting for the Maumee River watershed and how that delay ended up going all the way to August. Turns out we had about 50% of the land area that just never got planted in 2019 because they just could not get out there in time to be able to get anything out and planted. But that had repercussions going to fertilizer applications. So using some surveys, we found that only 46% of the typical commercial fertilizer amounts were even sold in 2019, that manure application was maybe 15% of normal between March and May. And that implies that this 30% lower than expected dissolved phosphorus loads was really attributed to the application of phosphorus. Probably not so much how much is applied, because our soil test phosphorus wouldn't change, the phosphorus in the soil wouldn't change that much from year to year, but really how it was applied and then what happens to it right after application or within a few months after application. So this is really interesting because this is going to dictate, we're hoping it'll help us drive and understand how to manage the Maumee River and the agricultural fields there moving forward. So hopefully this is where your brain is going right now and you're thinking, well, why has dissolved phosphorus increased? Didn't talk about that yet at all. And it's a complicated and hard to answer question that I don't even know if we have really perfectly right yet, but we have a lot of really good ideas. So in the 80s and 90s, the big push was to save your phosphorus and keep it on the field. So what you can see in this image here is that we have really intensive tillage on one side of the road here. And then we have this high residue conservation no-till management on the other side. And so the goal in the 80s and 90s was to go from seeing most of the landscape like this mulberry plow to most of it being more like conservation tillage with high residue management. And so that was really fairly successful, although you can still see some people still do this inversion tillage, otherwise I couldn't have gotten this picture. And that's what leads to, like I said, this big decrease that we see in total particulate phosphorus. What's intriguing about this decrease in phosphorus is that on the backside of it is we're seeing increases in flow, which usually increases erosion. I think if we did a correction for that over the past 15 years, we probably see farther, even farther declines in particulate phosphorus. So this was successful on a watershed scale. You never hear that, that's great. But one of the things that can happen if you move to a no-till type management, especially with residue, is that we start to get increases in dissolved phosphorus. So this is a paired field study over, a long period of time. You can see that, you can see that the conventional tillage was maintained in one of the fields and no-till was implemented in the other. And you see this big increase in dissolved phosphorus concentrations in runoff, and basically it stayed high. So this is pretty concerning, right? Like, seems like that's what happened. But one of the conundrums has been that most of our farms are actually rotational no-till. So there's usually some ground working over that period of time. It's not, and none of them, very few of them are long-term no-till, which is what this was. And so the question had kind of been, well, can this really happen in this type of scenario? Although I think the expectation was that it wouldn't have, but I would say that it looks like maybe so. This is further complicated because most of this area has a lot of subsurface tile drainage. And basically this is because all of Northwest Ohio used to be the Great Black Swamp. It was essentially the receding of Lake Erie left a very thick, dense lake plain that's super flat with lots of clay materials that just really led to a great swamp in that area. No one could live there or do anything with it until it was drained. And so that was really popular for a long time and still is. Where essentially the way this works is perforated drainage pipe is put about 1 meter below the soil that lowers the water table so that you can have farming. What we've been finding is that the more systematic this drainage tile is, like you can see in this bottom right picture, the better yield that farmers usually get because they don't have some of these issues with really wet ground. You can see that typically what we have are these big long ditch sections with lots and lots of tile drains coming in. So what's confusing about this in part is that typically you have these no-till problems and it's associated with some sort of a surface runoff, but typically with subsurface drainage tiles, if a lot of flow is going through that, that is filtered through the soils. So what we think is happening, and we have some support for this, so I'm not going to be able to show you all of it, otherwise I'll be here for another hour probably, is a complication of a mixture of things. So what we think has happened is that we've had the development of what we call soil stratification in our ag fields. Basically what that means is that phosphorus is accumulated on that top, 5 centimeters, 2 to 5 centimeters of soil, right? So that's all the phosphorus is right there on the top. And this is happening for 2 reasons. First of all, if you're not mixing the soil, it can sit up there. There's a lot of residue on the top because of that as well. And then on addition, most of our fertilizer is applied as broadcast pellets in the fall. That's not very heavily incorporated, if at all. However, if everything was operating perfectly, this water, then when it rained, it would slowly filter through the matrix of the soil and filter all that out, and it wouldn't matter. And we don't, and then having tile drainage prevent some of that surface runoff. But what we think is happening instead, and what we have some evidence for, is that instead it rains, grabs all that phosphorus that's accumulated at the top, and it hits a macropore. So we're having things like cracks and fissures, earthworm channels, and root holes, and all those sorts of things, which those the earthworm channels and root channels like to usually be right above the tile drains because it's a little bit of area of aeration, right? But those are happening and they're creating these highways for any of that water that's on the surface to go right down into the tile drain and pop right out. There's a researcher who's doing a lot of work on edge of field research and is finding over 80% of our dissolved phosphorus loads are coming through subsurface tile drainage and not coming off the surface at all. So this is what we think is going on. And we're trying to gather up a little bit more evidence. We know that there's a lot of phosphorus stratification in our fields. We've done measurements of that. It's just linking together how you're measuring preferential flow through macropores is a complicated feature. So then, if this is what's going on altogether, what practices should reduce dissolved phosphorus? And right now, this is just the diagram that's the 4 R's of nutrient management. So we've got applying fertilizers using the right source, making sure you're accounting for everything that you're applying, right? Putting it on at the right rate. So you test your soil, don't over apply. Putting on at the right time. So not on frozen or snow covered ground, not before a rain event. And then putting it on the right place. And that is basically not putting it on the soil surface if you can help it. And what it seems like is maybe some combination of these 2 things is the most important. I still think that right rate's important because we need to be looking at our soil tests to make sure we're not over applying. But I would say between 2019 and what we think is going on with these combination of macropores and subsurface drainage, that placement is going to be really key. Trying to get that phosphorus off the surface to prevent this development of stratification seems to be the thing that we need to pay more attention to. There's some evidence, there's been some research done looking at this, looking at, well, if we injected phosphorus fertilizer, how effective would it be at lowering DRP concentrations? And you can see in this figure, this was used lysimeters in the soil. So basically doing a simulation of rainfall and seeing what's basically coming out the bottom. And what you can see here is that broadcasting had the highest DRP concentrations, but injecting and tilling, that sort of inversion tilling, were about the same. What that implies is that no-till, the issues with no-till don't have to do with the fact that we're not tilling, but rather it has to do with phosphorus application. And we might be able to find that sweet spot where we can still inject phosphorus and get the reductions we need and have these reductions, and get the reductions in phosphorus. So we won't have, we'll have healthy soil and less dissolved phosphorus loss. So I know that you guys probably have some livestock issues as well, perhaps, and you might be thinking about, well, what about manure? I mean, you haven't talked at all about that. And the reason is because in Lake Erie, our soil test levels are not all that high, and we have maybe of all the phosphorus coming into the Maumee, it's somewhere around 10 to 15. Well, I think I've seen estimates as low as 10% and as high as 30% of the phosphorus, which means the vast majority of the phosphorus is from commercial fertilizer. And the estimates for the amount of land covered in by livestock or with manure applications is something like 7 to 10%. So manure is not the driving factor most likely in the Maumee River, but we do have other sites that is the main issue. So I would like to welcome you to Grand Lake St. Mary's. Grand Lake St. Mary's, as I mentioned before, is Ohio's largest inland reservoir. We have been monitoring around there. We have one creek we've been monitoring as an inflow since 2008. The other two have been after that. And you can see this is a picture from the Ohio Traveler. So it's a tourist magazine. This is before they realized how bad the issues were of them promoting going or canoeing on this lake. And you can just see how green it is. It's incredibly eutrophic. And so what ended up happening is that in around 2007, our EPA did a national lake survey. And with this national lake survey, they just basically collected samples of microcystin toxin and chlorophyll A, so how green it is, from lakes all over the nation. And Grand Lake St. Mary's came up as the 3rd most toxic and in the 97th percentile for chlorophyll A concentrations. And then basically everything started going downhill for Grand Lake since then. What we know is that this watershed, and you can't tell right here, but the watershed is basically just south of the lake, has basically huge amounts of livestock. That's where most of our confined animal feeding operations are. In Ohio, they all tend to be a lot right there. And traditionally, the advice for dealing with manure has been, you know, it's not going to go anywhere from farm fields, so just keep applying it. So we have some really, really high soil test phosphorus levels, like over 500 ppm of malic, if you think about those very often. And because of that, we have this sort of legacy losses of phosphorus. So in 2011, Ohio classified this watershed as a distressed watershed. Following that, 99% of the livestock producers, and therefore also 90% of the cropland, ended up having a nutrient management plan. It's not very regulatory, but at least says, let's test the soils and figure out, like, you know, how much you can really apply and what you're going to do if you have excess manure. That excess manure is basically shipped out of the watershed and used elsewhere. 20% of the watershed has received funding for cover crops, but I'm going to admit this is kind of old now, and I know that it's most likely that that has not been well sustained, but maybe it has. And then in 2013, they implemented a manure ban period. So there's no manure application allowed between December 15th all the way to March 1st. There is a loophole, you know, you can get approval if there's going to be some sort of emergency, because you don't want to accidentally release manure all the way into a creek and have it kill a lot of biota. So I'm going to focus primarily on Chickasaw Creek because we started that in 2008. So we have a good before and after manure ban period. And what I want to do is just show you some of our data that we have over time and then associated with those potential changes in distressed watershed rules. So here's our data. This only goes to 2016 because this is when we decided to do this study. It was basically following 2016. And at that time, we were starting to see some really, you know, it seems to be pretty good downward trends in our nutrient concentrations. And you can see that in total phosphorus, nitrate, total Kaldol nitrogen, which is basically particulate nitrogen and some ammonium. And then our dissolved phosphorus is not quite as much of a decline. Probably where we've seen the biggest decrease in concentrations has been in suspended solids. So just, you know, mud in the water. Those have gone down by about 2/3, which is really impressive. However, it takes a lot of years to get enough data to say something is really a substantial, real trend. And so we wanted to look at this data in a slightly different way than we have with some of our other really long-term data. So what we did is we separated each one of these into different flow categories and before and after the manure ban to answer two questions. We wanted to see how these concentrations, sediment nutrient concentrations, changed with time, and how did it relate with this manure ban. And then I have to show you probably the most complicated figure that I've had to show you yet. But basically, here are all of the ways in which we classified the data to answer these questions. So we have before and after manure ban, that's what the pre and post is. We have 3 flow categories because we know concentrations change with flow. We wanted to see how these concentrations responded under these different conditions. So we have a low flow, medium, and high flow category. And then we have during the period of time in which we have the manure ban and during the period of time in which we don't. So is it from December to March 1st, or is it March 1st to December 15th? Okay, laying that out, let's see some arrows, because that's fun. We have dissolved and white and particulate and black. And so what we found was that going from before when we had these manure ban regulation and to after, both in terms of dissolved and particulate phosphorus, we saw decreases in concentrations no matter whether we were at low, medium, or high flow. So you can see that these are some pretty good decreases. The decreases were not as strong or obvious under these high flow periods, but nonetheless, they were still there. And using the very complicated statistics model that my collaborator used, we found out that they're all significant. If we look at our non-regulatory period, it gets a little bit more tricky, right? So we had some decreases or very little changes, particularly at low flow or at high flow for particulate phosphorus. But for dissolved phosphorus at high flow, not really much change at all. But then we have this weird one, right? So you have this increase during medium flow in dissolved phosphorus during the non-regulatory period. So we had to dig into that a little bit and found that in fact what was happening was we get to March 1st and then everyone would be like, oh great, now we can apply our manure, it'll be fine. And so a lot of application happened that happened to be mostly within that medium flow category, because during high flow it was hard to get on in fields. So you're able to get out and basically have this mass application manure. We saw this bit of an increase in phosphorus. So pluses and minuses here in terms of how effective this has been. But where it's really been effective is in nitrate nitrogen. So you can see basically under both medium and high flow periods, whether you're during the ban periods or not, we saw decreases from before to after these rules went into place. And some pretty big decreases too. Like this was up around 18 milligrams per liter, and now we're down to more like 11 or 12. As a note, 10 is the drinking water standard for nitrate in the US. But we didn't see as strong a change during low flow. Concentrations are a lot lower then. That's when it's really hard to affect any change anyway. However, I want to give you some perspective here. So it looks like we had these huge changes and oh my goodness, Green Lake must be fixed. And this is something that was really important for us to actually get across to some of our regulators, because we didn't want them to stop trying here, right? Is that if we look across all of our sites and where we were and where we come to, the trends are definitely there and they're impressive, but we have a long ways to go. So this is showing total phosphorus, dissolved phosphorus, and nitrate. And we have basically almost all of our sites here. These middle sites are some Ohio River and Lake Erie on the front end. And basically what you see in Chickasaw Creek is that for total phosphorus, that's a pretty good decrease. Dissolved phosphorus, the decrease has been far more minor. But nitrate, I think, had the biggest decrease in concentrations over time. But look how far it has to go. Like, we have a long ways to go, at least in dissolved phosphorus and nitrate, just to be at where we are in Lake Erie. And we think Lake Erie needs to be decreased by like at least 40%. So if that's our case, then there's a long ways yet to go in Green Lake St. Mary's. I would say the fact that it seems like we need to keep doing what we're doing to deal with our particulates and our nitrate, but for dissolved phosphorus, I think that we need to start considering other alternatives for reducing that, because it's probably going to come off these fields for a long time. And that might include things like phosphorus filters or phosphorus traps at the edge of field. So here are some recommendations. We're going to have plenty of time for questions. I think I talked too quickly in the beginning. I apologize. But here are some recommendations. The first one is that I'm hoping you get a takeaway is that sometimes we find that BMPs come with trade-offs. It happens. Sometimes we don't think those trade-offs are going to be that big, and then do it on a watershed scale. And next thing you know, controlling for erosion leads to an unintended increase in dissolved phosphorus, right? These are things that can happen, and I would just say we need to make sure that we understand these trade-offs before going all in on any one BMP, right? The second thing is that not one BMP has to fix like everything. We don't have to have one silver bullet, but you wouldn't know that if you talk to a lot of the people on the ground. So BMPs should be combined if we want to reach these multiple goals. In our area, it's all cover crops. Cover crops are going to be the solution to all things, but it might not fix everything. There's some studies showing that it doesn't do a great job at dealing with dissolved phosphorus, especially up where you guys are, where you have like these big, really intense winters and then snowmelt periods. Usually any residue on the top leads to even more dissolved phosphorus runoff. So it might not fix everything, but it could fix some things that would be really good, like improving soil health and water infiltration and those types of things that can buffer that farm against other issues like climate change that could affect yield and bottom line profits, right? Doesn't have to fix everything. The other thing is, like, for instance, I showed that combined sewer overflows is a fairly small proportion of the phosphorus in the Maumee River. Okay, that doesn't mean that we shouldn't control combined sewer overflows, because, you know, they're pretty gross because they provide all this E. coli that comes into our waterway that's bad for humans too. So you know what, maybe we should also still deal with combined sewer overflows as well as do all these other BMPs so that we can do lots of things to improve our water quality all at once. So it's not all blooms. The last thing is that the solutions and the expected recovery time is probably going to depend on what type of system you're looking at. The Western Basin in Lake Erie responds really quickly to reductions in external nutrient loads, right? Sorry, of course, now I get a tickle in my throat. But anyway, if we have a drought year, so we don't have that many loads coming from the Maumee River, Lake Erie doesn't have a bloom. It doesn't have this like huge accumulation of phosphorus in the bottom of the lake that leads to a bloom no matter what. That's really great for Lake Erie, although obviously we're having challenges trying to reduce those external nutrient loads in normally wet years. But at Grand Lake St. Mary's, where we've had over application of manure for a really long time and there's a lot of phosphorus in the soil, we're probably going to take a long time before we get to those big reductions. What's great is that we're actually seeing some changes in a fairly short time frame, but we have to know that there's still a long ways to go before we actually get to where we need to be. Grand Lake, in addition, is a much smaller reservoir. It has a residence time of like sometimes over a year. So you can imagine that it might take multiple years just for whatever's in that system to come out. They might have to even, it's a reservoir, so they might have to consider doing some dredging if we get to the point where the water quality coming into the lake is actually substantially improved. So with that, I would like to throw up my contact info. If you have any questions or you need any other information and you don't want to answer them here, you want to send me an email, please feel free to do that, or follow us on Facebook and see what we're up to. And I think I can answer some questions if you guys have any.
Yeah, thank you so much for your presentation. That was super engaging, and I don't think you spoke too quickly at all. Janine from Alberta Environment and Parks will help us to facilitate some of the questions. So Janine, I think there's a couple in the chat there.
Yeah, you bet. There's a few in there already. And if folks have questions, please feel free to type them into the question and answer function. So the first one that we have is there any work being done on the policy side about timing of fertilizer application in the Erie watershed?
Yeah, there is. And it depends on what state you're in and, well, what country you're in, honestly, because, you know, half of the lake is Canada too. On the US side, in Ohio, we actually have rules against application of manure before rain events or on frozen or snow covered ground. So, and I was supposed to mention that I think that weird instance where we saw that increase in phosphorus because of probably not great timing and application of manure most likely won't be happening all that much anymore, because there's been additional rules put in place since then where that's banned. And that goes for both commercial fertilizer as well as manure. So it's banning both of those, which is pretty, I don't know, that's pretty good policy to me. You don't see that, and I don't know if you see that exactly that strict of a rule up in Michigan. And I honestly don't know if they have similar restrictions in Canada, but usually Canada is a bit more progressive. At least Quebec is a little bit more progressive when it comes to those types of rules. So my guess would be yes.
Okay, great, thanks. And I think Brad has a question for you as well.
Okay.
Yeah, can you talk about some of the parameters that go into that bloom severity or bloom size index for Lake Erie? And is that a model that's specifically calibrated for Lake Erie, or is it applicable at other lakes as well?
Yeah, so I'll just go back to that slide so you can see it again. I don't have it in better detail than this, so we're just going to have to go with it. Okay, so essentially you can see these two satellite images here have like different colors to them. And what ends up happening is the satellite itself is looking at essentially the spectra of what's coming back to it. So it's able to say, okay, well, we've got both an aerial extent as well as an intensity of the cyanobacteria where it is. So you can see like these pink ones here, like super high concentrated cyanobacteria. And so basically it's a combination of those two factors that makes up this index. And so that's how they get to that one number. So we take this cyanobacterial index and we put it on a scale. It's almost a log scale of 1 to 10. So people can be like, 10's bad and 1's good, right? Just to kind of give some reference for folks. The actual model for the bloom forecast is remarkably simple. And it is essentially this equation that you see in front of me, but a little bit more complicated because the NOAA scientist who does it, leaves it sort of in its more nonlinear phase where I've put on a log scale here. But basically you can see that the relationship's pretty straightforward. We have some other estimates that we use to help constrain what the model would give us. So we have like, we use both a flow model as well as a total phosphorus model. But as you can tell, like in 2019, we focused way more heavily on the bioavailable one because we knew of these other issues we were having. There is an ensemble forecast that goes into this. So while NOAA we do early season projections working really closely with them, the ensemble forecast actually includes other models as well. So there's a Bayesian model that comes out of University of Michigan. It uses a lot of the same input variables, but obviously a very different statistical background to it. And then Limnitech has a model. So that's a consulting firm in Michigan, and they have developed a dynamic ecosystem model. So it has got all the input variables of everything in the lake, like the current, using hydrodynamic models to talk about, how everything's moving, and it has all the inputs from all of the watersheds around, including Maumee, to be able to develop what they have. And all of that comes in to do this one forecast. So it's like, essentially, I'm going to move forward a little bit here where you can see each one of the little different dots. And that's, each one of those is just a different model. Did that answer everything?
Yeah, I think that's really interesting. We've been tracking blooms using satellite imagery on one of our major recreational lakes this summer that's prone to harmful algal blooms. So it's interesting to see kind of the end product that you can get out of tracking it with satellites.
Yeah, and I guess I should specify that, what he ends up doing is taking, for any given year, it's like he's taking the 10-day average during like the most intense period of time when there was the bloom. So like, that takes time for the bloom to form. It usually starts in the middle of July, and then it gets really big by August or September, depending on the year. And then if it cools off, it's been cooling off a lot here lately, then it sort of dies down. It can persist all the way to October, but not always. So it does incorporate, some of those longer periods of time. But the other thing that you mentioned is like this particular forecast is really unique to Lake Erie because it's using our actual monitoring data as input variables. And I don't know that, I think in other lakes it could be really difficult to forecast bloom size, but using satellites, you can at least, if you had that and some idea of current, you could probably forecast at least location.
Okay, great. Thanks, Laura. We have another question for you about blooms in general. So they're wondering about the work that was done to exclude light and temperature changes over time as potential co-drivers of blooms in Lake Erie.
Yeah, so, we would look at water temperature over time, and there hasn't been any real drastic changes. Like, I think there's a slight increase, but it hasn't been enough to change the bloom size all that much quite yet. That could be something that becomes a factor as we move forward. There's been a couple of really interesting years. The one of them is 2018. In 2018, it got really warm really early in the year. And so we actually saw the bloom kind of start to form in June at the same time as other things that were not harmful were blooming. And so then that bloom, as you can see, it was fairly not very big of a bloom. It ended up being a little bit lower than expected, when we think it was in part because it started under this competition with a lot of other organisms for nutrients. And then on the back end of the season, it started being like super windy and chilly. We got these big storm events that mix everything up, and that's really bad for microcystis. So then it's like, it was already sort of suffering on that other end. We have had other years. So another example is like in 2015, we actually didn't do a great job of forecasting that bloom. We didn't think it was going to be as big as it was going to be. And that was because we got a lot of rain in July. And before 2015, we didn't think that July mattered that much, because in 2003, we had a lot of rain in July. And look how little 2003 is. Well, it turns out in 2003, it was cold through like July, like below normal temperatures all the way through July. And so the whole bloom formation was offset a little bit. And so we, are like, oh, well, clearly July matters. It's just, it didn't matter so much in 2003. So we know temperature, especially those types of dynamics in temperature, can have an effect. It's just they haven't been consistent enough for us to see those changes. For light, that's never really an issue for us. It's, if it's summertime and it's a Great Lake, there's plenty of light. The only time there's a light issue, I should bring that up, is if we have a lot of turbidity. So if we have like those July storms, sometimes bring in a lot of mud. And so it can prevent a bloom from forming in one area, but usually it's still forming in other areas.
Okay, great, thanks. And one more question here. Has there been any work done on how snowpack from the previous year impacts loading the following spring?
Yeah, so not much, because we don't really get much snowpack. Like last year it didn't snow. So maybe I think we got an inch in April, which was like the exact wrong time, because we were in quarantine, and it's like, all I want to do is be outside, and now it's snowing, which is wonderful. But yeah, so not a whole lot, but I think there's something really interesting there, because some of the years we have slightly higher than expected concentrations in March, right? So it's like right when we're doing that sort of freeze thaw sort of period, we get like this big spike in phosphorus in March, and that usually follows an especially cold February. So when we were down, in Fahrenheit, when we're down in the like 10 degrees, so it's probably like -10 Celsius, right? When we're down where it's like pretty hard, really solid soil is frozen, in those scenarios, we start to see some of that runoff. Could be that there was some not really allowed surface applications that were going on that, is that runoff. But I also think there's some sort of interesting thing happening when you have high residue and that sort of thawing period where only the surface is thawed and then like the whole bottom is still really solid. And so you get like a little bit more surface runoff than you would normally get, where normally we get subsurface. Yeah.
Okay, great, thank you. That's all the questions that we have that came in.
Awesome.
Could you maybe mention with the frequency of your sampling, it's amazing how frequent you're collecting that data from those tributaries. 3 times a day, I think you said that it's being collected. So that seems like on one extreme side of the scale where you have really frequent sampling. Is there kind of a sweet spot where maybe we don't need to have 3 times a day, but obviously more is better, but I'm just curious about why 3 times a day is kind of chosen.
Yeah, so actually, and I didn't clarify because usually it's a lot of detail, but technically what we do is we analyze all 3 samples if the flow is up, otherwise we analyze 1 a day. So it ends up being about 500 samples per river per year. And we're doing that because, well, so imagine we started in 1975-ish and we needed to calculate, we wanted to have like the best estimate of loads. But some of the statistical power and the statistical techniques to be able to get that and fill in missing data, but they basically just didn't even exist. Or if they did, I know for sure we were using Fortran code on cards that we had to take to like supercomputer sort of things to be able to run those, right? So it's like, it was hard to get those numbers if you had to figure that out. So the more you calculate, the less you have to, you measure, the less you have to estimate. We've done a lot of studies since we have such a high frequency sampling. We can take samples away and see how much do you really need. And you're exactly right. So It depends on what your exact goal is. If you want really good loading numbers, we analyze far too many daily samples than you really need. All you really need is the really good storms, because if you get the monthly samples, right, at low flow, and you basically capture every storm, you know, it doesn't 3 a day, whatever makes sense, maybe 1 a day if you have a really big river, maybe more often if it's a really small river, you know, that basically gets us the same estimate, because all of the change happens. The load is the concentration times flow. So the load is all at those really, really high flows. If, let's say instead you're like, I want to know if an area might be a problem area. I don't care if I know the very exact perfect loading number. I just want to know, do we need to focus here? I think you could do some really savvy coordinated sampling during particular periods of time and just be like, okay, well, we're going to sample every day in May, and we're going to get those 30 samples. And if it rains, we'll get it. If it doesn't rain, well, that's unfortunate, right? But like, you know, you would hope that you would pick a sort of rainy period, or if you guys are dealing with snowmelt, be like, look, I know this is our snowmelt period. That's when all our load's going to happen. Let's sample for this period of time, and maybe just make sure you're just really consistent. And I think you'd get an awful lot of really good information from something like that. If you can't do that, my other strategy has been like just picking a couple of storms in each major season. For us, that ends up being something like early spring before we get runoff, but before there's any crops on the ground. Then sort of like the summertime when there's crops on the ground, we still have runoff. And then like that late fall, early winter, after everything's been harvested, it was super dry, and then stuff is just coming back, right? Those are the times that I'm like, if I get 2 good storms for each of those, then you can start to do some good relationships there, looking at like concentration versus flow and how that changes with different, different streams. So, yeah.
Yeah, that's good to know, because it can be intimidating seeing such an intensive project and thinking like we need that level of intensity. But so that's really helpful to kind of contextualize it.
Yeah, because you could then compare the concentrations of the different things you're looking at and be like, well, you know, you could have it over time. You can see, you know, have an understanding of your trends, or at least be like, well, do I need to be worried about this concentration versus that one? And, you know, start to make some changes.
Yeah, that's great. And I think you showed a website at the end of your presentation, Lake Erie algae or something like this.
Yeah, I can put it in the chat. The Lake Erie algae website, it's a website that we created. We had some help at the end of a project with some media folks to sort of tell the story of what was going on in Lake Erie and how our data play a role in it. So I stuck it in the chat if you guys are curious about it.
Perfect.
Oh, she found it too. Good job.
Just put it out to all attendees.
Yeah.
Okay.
Yeah, I was like, oh, I guess I can't do all that.
Yeah.
Oh no, I could. I'm just not smart enough.
That was a really great presentation. It's interesting to see, you know, something like what we do here in Alberta scaled up to the size of the Great Lake. And I think there's a lot of kind of lessons learned from your project. So I really, really appreciate you joining us today.
Yeah.
Great presentation. Thank you as well to our first speaker, Dr. Michael Rennie, who spoke about microplastics. These presentations are recorded, so they will be up on our website probably next week. And next Friday, we continue our series with a presentation on loon research in Canada and what that is telling us about lake health, and then following that up with some Eurasian water milfoil work out of the Okanagan Basin. So a bit of a different topic next week, so I hope you guys will join us next week. And thank you, of course, to Janine for helping to facilitate again today. Okay, thank you, everyone.